I'm Victoria Augoustides. I'm a senior in the bioprocess engineering concentration under the biological engineering major with a minor in biomanufacturing. I'm studying how pine bark can be transformed and valorized through different treatments with a catalyst, enzymes, and then a base, and then turned into biochar, and then evaluating the effects of each of those treatments on the ability of the biochar to act as a biological sponge on swine waste. Growers that are under a larger contractor are tasked with treating the waste and removing harmful contaminants from it before it can be, reintroduced either as a fertilizer or back into the soil. The technology that is currently used by growers is not new, and what we're trying to do is make it more efficient, make it so that the waste spends less time in the ponds and has less of an impact on the environment, thereby improving farm efficiency as well as just the general climate friendliness of animal agriculture here in North Carolina. We use pine bark and we run it through some water for a while just to wash off any debris and dirt, and then I dry that in an oven just to remove any water weight. So when we measure it out, it's what we're measuring the raw, dry biomass, not the water. Then it's ready to be run through its first treatment, which is a catalyst called niobium oxide. My colleague Autumn is trying to add value to lignin, but for me, unfortunately, it gets in the way of trying to get the cellulose and turning that into sugar. And so I remove that with a catalyst. And then the next stage, once that is sat at 100 RPM for 24 hours at a specific loading, we remove the catalyst, we wash the biomass off with water just so that it doesn't interfere, and we dry it again so we can weigh it out accurately. So it comes back out of the oven, and then we measure it out for enzymatic hydrolysis. Enzymatic hydrolysis is where we actually pull the sugars out from the biomass and turn cellulose into glucose. And so we're doing that, as I explained, to add value to the biomass and make this process more profitable. After it's done with the hydrolysis, I spin those down in order to separate the pine bark from the sugar and then run the sugar through an assay or a test to figure out how much sugar was actually produced in the process. The next step is making biochar. It goes in the furnace at very high temperatures, roughly at 1,100 degrees Fahrenheit. We flow nitrogen through there to remove any oxygen and prevent it from combusting. And so while it's burning in the oven at such a high temperature with no oxygen, all of the carbon-containing compounds are being burned away, such that all you have left now is a pure carbon skeleton. We treat it with KOH for 24 hours, and then we perform absorbance testing in several different treatments. At each step of the bioprocess, I'm taking out samples. So when we did the catalyst, I took out a sample. Before we did the hydrolysis, I took out a sample, and at each point, so that I can see across the bioprocess how the absorbance is changing, its ability to pull out p-cresol from the swine waste, or volatile organic compounds from the swine waste. What we're thinking of doing is putting the biochar in little mesh bags so it doesn't seep back out of the mesh, but it allows the liquid to flow through and over the biochar, and then allowing the biochar to pull out the chemicals from the swine waste. The other thing I think that makes this project specifically different is no one's investigated loblolly pine bark biomass at all. We recently published an article about the chemical characterization of loblolly pine bark, and it was the first of its kind just on the data that we published and characterizing it. If you want to explore a bit more, coming to a lab like the Waste Lab is what I would recommend, and just getting involved in as much as you can, helping other students out with their experiments, learning protocols, documenting things, learning how to keep a good lab notebook. Those are really good translational skills that don't just have to be applied in a research lab. And coming to a lab where science is applied rather than just you're looking for new things, I feel like is really beneficial for engineering students.